Welcome back, Bio 100 crew. No matter how you're consuming this media, I hope you're having a wonderful day. I hope you're feeling good. And I hope that the semester is kind of starting to hit a nice little groove for you. You know where things are, and you're feeling good about what's ahead. Please don't forget that we're here for you in our office hours and otherwise by appointment. If you need anything, all the instructors in this course are more than happy to help with whatever you might need. With that said, let's talk about what you're going to find here in video number 2 for this week. This is 3.2, data collection and analysis. That last part, the analysis, that's going to happen a little bit more throughout the next set of videos and questions, and even throughout the next couple of weeks. So this one is really going to be heavy on the data collection side and what you might do with a particular type of experimental data. Here, we're going to focus on enumeration of microbes in an unknown sample. And don't worry if you haven't taken microbiology. No advanced knowledge of anything related to micro is needed for this. What we're really going to do is put to work our powers of 10 and our dilutions idea and solve a complex problem using those ideas, which is to determine the concentration of microbes in a sample. We'll get into all the little details soon enough. But first, let's go over the overall learning outcomes for this particular module. First, because we're going to use our powers of 10 and our rules of dilutions, let's go ahead and review those rules. Whether you took Bio 2 here or whether you've transferred in and taken an equivalent, this particular video will catch everybody up and make sure we're all on the same page as we head into class this week and looking forward. The second thing we want to do is review or teach the rules for countable colony range, and then we're going to go ahead and set up a tenfold dilution series. It's really important right away to think about this. If you have a sample at high concentration, you might need to do some dilutions in order to determine the concentration in that sample. We'll get there. The third is to, as mentioned, use all of this knowledge to find out what the concentration is. That sample might be soil collected from the dirt by the American River. It might be the amount of bacteria that are coming out of a milliliter of your spit. It could even be the content in your poop. We'll get there in a second. So thanks again for being here, and let's get started. So as mentioned, you don't need to be a microbiologist to complete this particular video and questions. But I do think it's worthwhile to get us together about a particular concept just so that you have some context for what we're going to do. And so it's important to remember that the reason we're doing this, meaning determining concentration of microbes in a particular sample and using our mathematics to achieve that, is that microbes are everywhere. We're surrounded by them in the environment, on our skin. They are literally everywhere. We cohabitate this planet with them. I suggest that we are codependent on them and we have co-evolved with them. And they're often in existence at high concentrations. So that's an important thing to consider when you want to enumerate them. As I even hinted at in the first slide, they're in your poop. In fact, some estimates suggest that up to 80% by weight of every poop stool you create is the bacteria that are in your gut getting passed out with those stools. And so what's in there? What's really in that poop emoji that is supposedly a chocolate ice cream cone? I'm not buying that. I think that's a poop. You might find yeast and fungal species and bacteria and viruses and mites, all those things, okay? That's just going to set the stage. It's not important that you're taking notes about this. It's just to give you the idea that they're present, that they exist in high amounts, and that it's often important to determine how much are in there. And we're going to get into that particular component during the in-class exercise for this week, when we take a look at how bacteria recovered from a mouse might be affected by one treatment or another treatment. And that's a really important thing when you think of human disease, for those of you who maybe down the line are going into some sort of biomedical related science or even an environmental related science. You might be tempted to think, I'm not sure I'm covered by bacteria, and it seems here that even Beyonce isn't convinced. But I'm telling you, even Beyonce, the great Beyonce, is covered with microbes, trillions of cells, doing really, really important things for her normal physiology, keeping everything in balance. I thought about putting a little picture of a glass of lemonade on the slide, but that's a little too heavy-handed. Let's keep going. Now that we're maybe a little bit more comfortable with the idea that it's okay to have microbes all over your body, and it is, the question now becomes, what if I wanted to quantify them? How would I actually go about doing that? And that's where we're headed. Let's think about this for a moment. If we took a sample, let's say of Beyonce's poop, and we wanted to know how many bacteria were in there, we could create a little slurry of it in a sterile solution, and then we could take an aliquot, a little bit of that, and pipette it onto a solid agar plate. And you've seen those before. Those are growth medium for bacteria. But if we did that without diluting the sample first, all we would get is what was called a lawn, a complete covering of bacteria on the plate. There's nothing countable there. To determine the amount, we need to count colonies, and therefore we need to do dilutions, because we've already said that, hey, microbes are everywhere and they're often at high concentrations. And so that rule of thumb is where we start. Our goal is to dilute as far enough out into sterile medium as it takes, and we're going to create tenfold dilutions to do so, such that we achieve at the end of that a countable colony range between 30 and 300 colonies, or CFU, which stands for colony forming units. Basically, how many colonies do you see on a plate of agar? Here's my little illustration to represent that. And if you want, you can feel free to do this with me. But I'm going to tell you right now, as a spoiler, there are 100 little colonies on that plate. So the colonies are represented by the little orange circles, and here they sit nice and pretty, all separated out beautifully on a yellowish solid agar plate. Again, this isn't microclass, but I do want to kind of define some terms just so you feel comfortable with what I'm talking about. And so each of those is a colony forming unit that you see over here on the plate, all 100 of them. Each one is technically called a single colony. And I just want you to know that a single colony is actually derived from a single cell. And so what you see here are 100 groups of bacterial cells, each of them starting off from a single cell. And there's a hint as to why we have to do dilutions. You have to get to such a low cell number such that when you put it on a plate and you grow them, each of those cells will grow into a colony that doesn't overlap with its neighbor. I hope that makes sense. We'll keep talking about it. A single colony, then, is a collection of cells. They're all genetically identical. Remember, bacteria cells are so small that an individual cell would need to be observed with a microscope. But you could see these plates with your naked eye. And so therefore, each colony represents a grouping of cells that all started from a single cell, and then by binary fission, double, 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 you get a collection of cells within each colony that are all genetically identical. And if you're also hungry for In-N-Out Burger right now because of how many times I just said double double, my apologies. So the last piece here is that a single colony is considered a pure culture. That'll be important in another class. That's another show. We don't need to think about that anymore for this point. It's only important in the context of go from a concentrated sample to this plate. How do we do that? If the sample's at high concentrations, we need to create dilutions first so that we get down to single cell level in the countable colony range of 30 to 300. And this plate, because it has 100 colonies, is definitely in that 30 to 300 range. Now that we're beginning to get a sense of why we would want to do this at all, let's go to the end and talk about how we would actually use a plate that looks like this to determine the concentration of a complex sample like bacteria in Beyonce's poop. All right, so units first. Units are always where I start any kind of calculation problem. I go ahead and write down the units first. Is it volume? Is it a concentration like it is in this case? It is. And so I need something per something. That is how we think about concentration. In this case, it is colony forming units, which we just talked about, per mil of sample. Or, in parentheses, I've written CFU per gram of sample, because in this case, we're going to be able to use the 1 mil equals 1 gram concept such that those are interchangeable. So CFU per mil and CFU per gram, we're going to consider to be able to use as the same. So let's go through this together. Don't panic, okay? In parentheses, I have here a division problem that represents the first part of how you would use colonies to get concentration of microbes in a sample. So this is your formula, essentially. So the first part is, in the numerator, the number of colonies that you found in the countable range. And again, that could be anywhere between 30 to 300. So there's where we get our CFU. That's going to be divided by the volume, which we added to the plate that you see at the left, that actually was used to get those countable colonies. So remember, we had to actually take a volume and spread it onto the plate, and then let it incubate and let it grow, and then those colonies formed. Whatever volume you plated on there that goes with those colony numbers is right here, and it's nice to have those in mils. We're going to multiply that by the number of tenfold dilutions that you used to get to the plate in the countable range. Maybe you did 6 dilutions, maybe you did 7, maybe you did 10. We're going to go through all of these things. I just want to give you the setup first, the equation first, okay? The tenfold dilution factors used to get that plate. So if you did 4 dilutions, that would be times 10 to the 4th. If you used 3 dilutions to get there, this would be times 10 to the 3rd. This will all become really clear as we go through the next couple of slides. Time to do some work. Let's actually go through an example and make sure we know what we're talking about, okay? So the first thing we do, and we're going to think about this like a lab experiment, so we're going to go step by step, is to obtain a sample. We need a sample. We want to determine a concentration of bacteria in a complex sample. We assume it's going to be at a high concentration. And if you're sick of talking about poop, well, maybe we've got Beyonce's saliva. Let's say you went to the concert, you were in the first row. I don't know how you afforded that, but that's a different story because those tickets are super expensive. Believe me, I looked. And then you got some of her spit when she was singing on your phone, the same phone you weren't supposed to have out, but you wanted to record the video you're not going to watch again that's on your phone unless it was confiscated, and you thought, wow, I have her spit, I want to preserve it, but I don't know how. Maybe I should preserve the microbes in the spit. That's a good idea, and while I'm at it, I could quantify it. Whew! If you're thinking that was crazy, yeah, it was. Here we go. Let's move forward. Those are the bacteria. I've colored them orange. And let's say in the lab, I took this complex sample and I have it in a sterile media, and It's darkly colored orange to represent the sample being at a very high concentration. The darker the color orange here means the greater the turbidity of the sample. If there's a high turbidity, well, that's just a way of saying indirectly, we sort of qualitatively speaking, that we have a very high concentration of microbes in that tube. This is our undiluted sample, and its value is 1. And I'm going to say that because you can go 1 over 10 to the power 0, which is 1. 10 to the power 0 is 1. 1 over 1 equals 1. To say that its dilution factor, well, hey, it's not diluted at all. This is the undiluted original sample. This is a value of 1, okay? We're going to go from 1, and we're going to keep making dilutions by powers of 10. So let's do that. On the right here, the rest of the slide, all this white space, is going to be devoted to making 10-fold dilutions. So underneath this gray bar, you're going to see a 10-fold dilution series come to life. So step 2 in the lab, we're going to grab our instrument of choice, and it's going to be used to transfer 1 milliliter of the homogenized sample from that orange original inoculum, and then we're going to transfer that 1 ml into a test tube, a new test tube, that contains 9 milliliters of a sterile medium. Okay, so transfer 1 ml into a tube containing 9 mLs. Let's see what that might look like. Okay, this arrow is to represent transferring 1 ml. Here's a new test tube. It originally had 9 mLs, and as you can see here, ooh, it looks a little less orange. I like that. Ooh, yeah, looks a little less orange. Maybe about a tenth less orange per se. Shall we? So, what do you mean by a tenth less? Well, let's think about that for a moment. If we took a ml of our concentrated sample and we added it to 9 mLs of sterile media, the total volume is now 10. Of that 10 mLs, how much of it is the microbes from the original sample? 1 ml. 1 over 10? 1 ml over 10 mLs? Well, that's a tenth. And that is why we call this a 10-fold dilution series. We just made our first 10-fold dilution, a tenth of what it was before. It is now a tenth less concentrated. And that becomes important to think about when you realize that your ultimate goal is to take an aliquot of this tube, or one of the tubes in the dilution series, put it on an agar plate, and get those single colonies. So I'm going to create 2 little rows, and we're going to put some numbers under each tube, and I hope this helps you think about what we're doing. So what is our overall dilution factor at this point for this particular tube? Well, we kind of already said it, but let's write it down. It's a tenth, because 1 ml of the sample is in a total volume of 10 mLs. One divided by 10 is 0.1. It's a tenth. And if you want to write it down a little differently, you can represent it as 1 divided by 10 to the power 1, or 1 divided by 10. Okay? And so you could see how that relates to the undiluted tube, where it was 1 divided by 10 to the 0. 10 to the 0, where 0 represented the original sample, and now 10 to the first, where the power 1 represents our first 10-fold dilution. Therefore, the overall concentration reduction in this tube now is 10 to the power 1. 10 to the first. This is the 10 to the first tube, because we are 10 times less concentrated than we were before. We have 10 times less concentration of Beyonce's spit bacteria. If we thought our original inoculum was a bit lower in concentration, maybe we could stop right there and take that aliquot we were talking about and put it on an agar plate and come back into the lab the next day and see how many colonies we have. But that's not the case here. We think we have a really high concentration to start with. So we need to make more dilutions. We need to make more 10-fold dilutions. So number 3 up there at the top. What we're going to do next in the lab is we're going to mix that tube up real nice, make sure it's nicely homogenized again. We're going to take a clean pipette or whatever instrument you're using to make your dilutions, and we're going to repeat this series over and over again, where you transfer 1 ml of the tenfold dilution into the next tube containing 9 ml of sterile media. You mix, you transfer, you mix, you transfer, etc., while changing instruments each time. Okay, so here's what that looks like. Remember, each arrow represents taking 1 ml and transferring it into 9 ml of sterile media, and our orange color is lessening just a little bit there. I want to stop for a second to make sure we're all on the same page, okay? See that little gray double arrow I just put in there? Between these two tubes, how different are we now? Well, we're a tenth apart, right? 1/10, or 0.1. We are a tenth apart, okay? Within a dilution series, each tube is 10 times apart. Again, why we call it a tenfold dilution series. It's a tenth of what it once was, and we're going to keep doing that, okay? But now, overall dilution factor underneath the tube. This is relative to not the tube just to the left, but to the original sample tube, because our goal is to have the concentration of that, okay? So now we've created 2 tenfold dilutions. Therefore, our overall dilution factor becomes 1 divided by 10 to the power of 2, 2 being our second tenfold dilution. Well, 10 to the second is 100. So another way to write this is 1 divided by 100. This is a hundredth of what it once was. This is a 1/100th of the original sample. Therefore, it's 0.01 in a decimal form of the original sample. What's the overall concentration reduction? You guessed it. I hope you're with me on this. 10 to the second. This is the 10 to the second dilution. It is 100 times less. Pause it there to think about that just for a second before we go forward. It's important that we're all on the same page. I know you can't ask me a question right now through the video interface. However, in class, we'll have plenty of opportunity to do this. And again, our office hours are available. We're happy to help. If we keep doing this, okay, if we keep saying to ourselves, hey, that tube is probably still too loaded with microbes. We need to keep diluting this thing. We won't get single colonies on a plate. It'll still be a crowded lawn. Let's keep going. Okay, orange color gets a little less as the green arrow represents moving 1 ml of the previous tube into 9 ml of sterile media. Okay, again, from the second to the third dilution tube, we are a tenth apart. We made another tenfold dilution, or 0.1 of what we were. When we think about this third tube relative to the original sample tube, well, our overall dilution factor, as you see underneath the tube, becomes 1/10 to the third, where the power 3 represents our third tenfold dilution. 10 to the third is 1,000, and that's 1 plus 3 zeros, so 1,000. 1 divided by 1,000 is 0.001, or taking 1 and moving it 3 decimal places to the left. And I hope that little throw-in is a flashback to the first video, and creating tenfold dilution series and thinking about powers of 10 is the same. Okay, so 10 to the third dilution tube, 3 decimal places to the left, that's 3 powers of 10, 0.001. Our overall concentration reduction is, as you were thinking, 10 to the third. And again, if you're not thinking, that's okay. We'll catch you up in class and in office hours. This is a 1,000 times less now tube at concentration than the original sample. So now we're getting somewhere. Let's keep going. Another green arrow. You guessed it, that's 1 ml. Here comes another test tube, looking a little less orange. And that was because we mixed it with 9 ml of diluent. This is the fourth tube, but if you look relative to the third tube, it's a tenth less, just like each of the previous examples. 0.1 apart. Overall, now this is the fourth tube, and therefore we can think of this relative to the original sample as 1 divided by 10 to the power 4, the fourth tenfold dilution. 1 and then 4 zeros. Hey, that's 10,000. So that's 1/10,000th of the original spit sample. And if we took 1 and we moved it 4 decimal places to the left, we get 0.0001. 3 zeros, 1. To the right of the decimal now. Okay? Relative to the overall concentration, it's important to keep this consistent. We now have a reduction of 10 to the fourth. This is the fourth tenfold dilution, or 10,000 times less. And because I have just a little bit of white space there on the right, let's do one more for fun. Let's transfer a mil. Oh, hey, I don't really see much orange at all. I don't see turbidity anymore at all. I think we're onto something. I think we're getting to a place where we could feel comfortable maybe taking some aliquots and spreading them onto agar plates. Relative to the tube before, this fifth tube is a 10th of the fourth tube. This is the fifth tenfold dilution, so its overall dilution factor is 1/10 to the fifth relative to the original sample. That is 1 divided by 100,000, or 0.00001. Sorry, I had to keep track of all those zeros. 0.00001. Or, relative to the original sample, an overall concentration reduction of 10 to the fifth, a 100,000 times less concentrated sample. Look at this picture. We took a concentrated sample of bacteria, and we thought to ourselves, we need to get to a countable colony range of 30 to 300. But we can't do it with this sample if we plated onto agar plates directly. We need to reduce the turbidity. We need to reduce the concentration. We need to reduce the numbers of bacterial cells in there. And we're going to do it by a 10-fold dilution series, where each tube has 10 times less than the one before. And we're going to do as many as it takes until we see very low turbidity. Now that we've created the dilution series, let's use it for our original goal, which was to determine concentration. What is the concentration of the microbes in that original sample? That's what we want to know. So, to do it, let's go through the setup. Transfer a mil from each dilution tube and spread it onto agar plates. Just to keep in mind, these volumes are for this particular experiment, but they could be changed. For example, all of your 10-fold dilution series, those could be in 1 mil volumes instead of 10 mil volumes. And all of your transfers could be 100 microliters instead of 1 mil. So this is just for us. We're going to start at the right and work our way back to the left. To the left, to the left. Irreplaceable, right? You're with me? Okay. We're going to go from the lowest density, the lowest concentration, and work our way back towards the highest concentration. So we're going to take a mil from that tube on the right. That's the 10 to the fifth dilution. We're going to spread a mil onto an agar plate. We're going to do the same for the 10 to the fourth dilution. We're going to take a mil. We're going to spread it onto an agar plate. And we're going to repeat this for each of the 10 to the third, 10 to the second, and even 10 to the first dilution, despite the fact that we think that some of these are going to have way too many colonies to count. They're going to be lawns. They're going to be smashes of bacteria all over the place. We can't count individual colonies. That's okay. Remember, we're only interested in one of these. We're only interested in one plate. We're going to disregard the rest of these. We want the one that's going to have 30 to 300 colonies. We want one plate. Only one of these plates is going to get the job done. Okay? So after we've spread our mil onto each of these, we're going to incubate in an incubator. We're going to do that typically overnight. And we're going to let those single cells we've spread on these plates grow on the agar medium into collections of cells called a what? That's right, called a pure culture, aka a colony, so that we can eventually count and enumerate CFU. So, from left to right, remember, we have 10 times less colonies that direction, just like we had 10 times less concentration. 10 times less concentration should correlate to 10 times less colonies, aka our 10-fold concentration reductions, aka our 10-fold dilution series, from left to right. It's worth a brief note here that we are working with perfect powers of 10 in assumption that there were no pipetting errors along the way. That's not quite true in practice. Between mechanical and human error in experimentation, it's never going to be quite 10, but you get the picture, okay? Lastly, the third bullet there on the left, we want to do what we came here to do. Calculate our CFU per mil, aka CFU per gram, of that original sample. As I mentioned, colony forming units per mil, concentration, is calculated from only one plate, the plate containing 30 to 300 colonies. That's important. To this point, as a quick little summary, we've taken a concentrated sample of bacteria that we wanted to know the concentration of from a orangey tube. We created 1, 2, 3, 4, 5 10-fold dilutions by taking a mil from one tube, adding it to 9 mils of a solvent, mixing, then transferring and repeating. Each of those tubes is a tenth apart. Each of those tubes represents a 10-fold reduction of concentration from the previous one until we have a power of reduction on the right of 10 to the fifth. 10 to the fifth less concentrated. Look at the orange colors decrease. Then we put a little bit of each, 1 mil to be precise, from each of those dilutions onto agar plates. We can look for the 30 to 300 colony plate and determine CFU per mil based on the formula we used and showed you in a couple of slides ago. So I hope you're ready for an example. Let's actually work with some numbers and do an example. And before we do, remember, there's only one number we're looking for, 30 to 300, okay? So let's do it. To make this all come together, let's do an example, in fact, where we actually know the concentration of the sample. To begin with, let's say we had some sort of Beyonce technology and we could determine how many bacteria were in there. We could know the concentration right off the bat, okay? Because if the concentration in the sample is known, then so too is the concentration in each of those 10-fold dilution tubes. That would make sense, with each being a 10 times less than the previous one. So let's go through this, making a 10-fold dilution series where instead of just abstract, how does this work, we use a real concentration and dilute it out. Here we go. Hey, same microbes. Hey, all right, same orange tube, good. Nothing new here. We need to determine that concentration, but this example says we already know what it is, okay? This is going to be really helpful when we determine how many dilutions we need to do and which plate will have 30 to 300 colonies. That's where we can really use the power of this, okay? So this is our undiluted, but in this case, I'm going to give you the concentration right off the bat. And we're going to do concentration here as CFU per gram, okay? And I'm telling you, technology is great here in this year, and we put it to work, and we already know that there's 1 times 10 to the 6th CFU per gram. And so don't turn the video off just because you know the concentration. This is about putting it to work so you understand the setup when you don't, okay? So if we have 1 times 10 to the 6th CFU per gram in this tube, and we take a mil and we put it in another test tube that has 9 mils of sterile media, that's our first 10-fold dilution. What's the concentration of this tube. What's the concentration of bacteria in this tube? Pause it for a second. Okay, welcome back. Let's use our handy-dandy chart to reveal that the overall dilution factor is still the same, 1 over 1 power of 10, and so a tenth, so 0.1. This is our first tenfold dilution series, and our overall concentration is, okay, good, just like before. Hey, this is going to be 10 times less, and because it's 10 times less, the concentration in this tube is 1 times 10 to the 5th CFU per gram. Compare that to the original sample, where it's 1 times 10 to the 6th CFU per gram. Do you see how 1 times 10 to the 6th is 10 times more than 1 times 10 to the 5th? And 1 times 10 to the 5th is 10 times less than 1 times 10 to the 6th, okay? In fact, it's 1 power of 10 less, 10 times less. 1 power of 10 is 10. So I took 6 and I subtracted 1 and I got 5, and that's where 1 times 10 to the 5th CFU per grams is. So we know our concentration in our first dilution tube. Let's keep going. Hopefully this will start to add up in your mind. If we do another mill into another 9 mills, oh, hey, let's remind ourselves, how far apart are those? If you said a tenth or 0.1 apart, you are correct. The overall dilution factor, meaning, again, from the original sample, is now 1 over 10 to the second. This is our 10 to the second dilution. It is 100th of what it once was. And if you're following along, you'll now know that the overall concentration reduction is, just as it was before, 100 times less, 10 to the second less than the original tube by creating these two tenfold dilution series. From the start to now, we're looking at 1 times 10 to the 4th CFU per gram. That's 100 times less than the original sample. It's 10 times less than the dilution to the left. The first dilution being 10 to the 5th and the second being 10 to the 4th. Good. Let's keep going. Let's finish this out. We did 5 tubes before. Let's keep going. Take another mill. Mix it with 9. Compared to the 10 to the second dilution, hey, it's a tenth apart. Good. Tenth less. Compared to the original tube, and therefore our overall dilution factor, we have 1,000th, 3 powers of 10 less, and that's 0.001. Our overall concentration reduction, therefore, is 1,000th of the original tube, 10 times less than the one to the left of it. And if you keep following along, this means it's 1 times 10 to the 3rd CFU per gram. We're not done. Let's keep going. We've got 2 more to do. A mill into 9, 10th apart. You guessed it, just like it was before. This is the 10 to the 4th dilution. It's 1 over 10,000, or 0.0001 of the original sample. The overall concentration has been reduced by 10,000 times, or 4 powers of 10, 6 powers of 10 in the original sample, minus 4 powers of 10 in this 4th dilution tube is 1 times 10 to the second CFU per gram, 6 minus 4. Awesome. And lastly, our 5th and final tenfold dilution is a tenth less than the 4th tube. It is a 1 over 100,000 times reduction from the original tube. It is the 10 to the 5th tube, and therefore 5 powers of 10, 5 test tubes in a tenfold dilution series less than the undiluted. So 10 to the 6th and 10 to the 5th, when you compare them, you see the difference there of 1 times 10 to the 1st CFU per gram. Therefore, if we knew the concentration to begin with, here 1 times 10 to the 6th, and we created these tenfold dilutions, we would actually know the concentration in each of those tubes. And that becomes super powerful when we think about that last step, which is actually taking volume from each of these and putting them into agar plates, because the concentration in each tube is going to tell us how many colony forming units we're going to get. And that is the next and important part of this video. So let's finish this work, because if we know the concentration in the original tube, then we know the concentration in each of our tenfold dilutions. Well, guess what? It turns out that if all of this is true, and our next step is to take a little bit of each of these and spread them onto an agar plate, well, guess what? We already know which plate will be in that countable colony range of 30 to 300 before we even throw those plates into the incubator. And you might not see that right yet. And that's okay, because that's the point of this particular slide. Let me show you how I can look at the concentration of my original sample, the concentrations of each of my tenfold dilutions, and know exactly how many colonies will grow on the plate. That's this slide, okay? And this is important when we're thinking about our experimental design. So as we went through last slide, our first tenfold dilution is a 10th overall concentration, and therefore 1 times 10 to the 5th CFU per gram, which is also the same as 1 times 10 to the 5th CFU per mil. That's our concentration in this tube. Okay, so our goal was then to take 1 milliliter from this tube, spread it onto an agar plate, put it into the incubator, and then come back the next day and see how many cells grew into how many colonies. Do you think this is the plate that has 30 to 300? Let's go through how. We know that actually the answer in this case is it's not. This is still way too concentrated. We really needed to make a lot of dilutions. This is going to end up in a lawn of bacteria, okay? Way too many CFUs. So we are looking for the number of CFUs on that plate. And I know the concentration in my tube and the volume I plated. So the question becomes, just like I said a few slides ago, is the colony forming units per the volume I put onto the plate. That was the numerator and denominator of our equation for this rule to determine concentration. And I'll put that together on the next slide. But here, if you took 1 times 10 to the 5th CFU in the numerator per 1 mil in the denominator, and you multiplied it by 1 mil, meaning the volume you spread on the plate, you would get mils canceling out, and you'd be left with CFU, so there's your units. And the math on that would be 1 times 10 to the 5th multiplied by 1, concentration up there by volume plated, and therefore when the mils are gone and the CFUs are left, you'd have 1 times 10 to the 5th colony forming units. You'd have 100,000 CFUs. 100,000 CFUs? Well, hey, that's way above the colony counting range of 30 to 300. Okay, so we're going to disregard this. Remember, we're only looking for one plate. Only one plate is going to be between 30 to 300, and 100,000 is not it. Before I even move forward, I want to ask you a question. 30 to 300 is the range. What's the number going to be? Not which tube, we'll get there, but what's that number going to be? What number is going to be between 30 to 300 if we're working from 1 times 10 to the 6th, we're doing tenfold dilutions, and we always have powers of 10 coming up. Pause that video for just a moment. Welcome back. I hope you were thinking about that and you realized that if you did these tenfold dilutions, they're always going to be 10 times less. We're starting from 1 times 10 to the 6th. Eventually, the only number possible to fall between 30 to 300 in our scheme is going to be 100. We're going to get 1,000, we're going to get 100, we're going to get 10, we're going to get 1. If we kept going, it would be 0.1 and 0.01 and 0.001. But there's only one number that's going to fall between 30 and 300 in this scenario, and that's going to be 100. So let's keep going, and I'll show you that. Our next tenfold, the second tenfold dilution, has 1 times 10 to the 4th CFU per gram, aka 1 times 10 to the 4th CFU per mil. And that is 10 times less than the tube next to it. And from the original sample, that 10 to the second dilution is 100-fold less, 100 times less, 2 powers of 10 less than the original tube, 6 minus 4. 6 minus 2 is 4 there. We're going to do the same thing. We're going to plate 1 mil. The question is, how many CFUs are in that 1 mil? What's the amount of CFUs in that 1 mil? Well, we know that the concentration is 1 times 10 to the 4th CFU per mil. So 1 times 10 to the 4th CFU per mil times 1 mil is 1 times 10 to the 4th CFU, or 10,000 colony forming units. That's 10 times less on the plate than the one to the left. However, that's way above the countable colony range of 30 to 300, so we're going to keep going, because that's not it. We're going to throw that one away. It's going to have a lawn, just a mess of bacteria on there. Well, how about this one? Okay. We're a little less turbid in the culture tube. You may remember its concentration is 1 times 10 to the 3rd CFU per gram, aka CFU per mil, and that is 1,000th of the original tube, 10 times less than the 10 to the 2nd dilution tube. This is the 10 to the 3rd dilution. I hope you're getting the language now here. And again, we're going to do this in class as well. So we'll all get to have hands-on interaction. We're going to spread a mil. And if you think about the number of CFUs that you're going to have in that 1 mil from that test tube, we're going to multiply that by the concentration to get the amount. Concentration times volume gives me amount, and I have 1,000 colonies. That's still too high. That's well above 30 to 300. We're going to look to our next dilution tube. This is the 4th dilution tube, the 10 to the 4th dilution. Okay, and as you can see, it's 4 powers of 10 less in concentration now at 1 times 10 to the 2nd. If I took a mil out of that tube and I plated it, the colony forming units I would get from the cells that I took out by multiplying the concentration times the volume, where 1 times 10 to the 2nd CFU per mil times the 1 mil I plated, that would end up with 10 times less than the plate to the left, and a number of 1 times 10 to the 2nd, which is 100. That is the magic number. There it is. That's the plate I've been after. That is 100 colony forming units. That is the magic one, the only one. This is the only possible plate from this dilution setup where I did 10 full dilutions, where I'm going to get countable colonies in the range of 30 to 300. Before I even do the rest of this, how many colonies are you going to get on the next plate? And if you said 1/10 of this, 10 times less, another power of 10, 1/10, it would be 1 times 10 to the 1st, which is 10 colonies, which is less than 30 to 300, you were correct. And so I'm going to finish this out, even though we know which plate we're interested in. If we took this last tube at 1 times 10 to the 1st colony forming units per mil, and you multiplied it by the 1 mil you plated, and mils cancel out, and you're left with CFU, you do indeed get 10 colonies, 1 times 10 to the 1st colony forming units. This is also now out of the countable range. This one is too low. This porridge is not right. There's only 1 bowl of porridge in this. The rest are too hot or too cold. The one that you want, and watch out for those bears. They're crazy. Goldilocks, you know what I'm talking about? All right. The only one you want is 100. And that came from the 1, 2, 3, 4th dilution tube. 100 CFUs. Outstanding. Let's do one more last thing to really make sure we understand. Okay, we've taken this example where we know the starting concentration, we took that and made 10-fold dilutions, and we knew the concentration, therefore, in each of the 10-fold dilution tubes. We took a mil of each and we put them on agar plates and we incubated them, and we could predict the number of colonies, and they were each 10 times less when you went from left to right, and we knew which one was the countable colony range, and we said that was going to be 100. Let's calculate backwards. Let's prove to ourselves that this is all correct. Let's do some backwards math. Let's actually calculate the CFU per mil, aka CFU per gram, in the original tube, just based on our original formula, as if we never knew. Let's pretend we didn't know it was 1 times 10 to the 6th CFU per gram. Let's pretend we didn't know. Let's pretend all we knew is that we made a 10-fold dilutions series from an unknown sample, and the 10 to the 4th dilution gave us 100 colonies. Let's put this all back in to our rule, where the number of colonies in the countable range divided by the volume added to the plate containing those countable colonies, multiplied by the 10-fold dilution factor for plates in that countable range, that one plate, gives you the CFU per mil or CFU per gram, the concentration. In our example, what was the number of colonies in the countable range? It was 100 CFUs. So in this case, we're pretending that we didn't know the concentration in the original tube and we're trying to find it. We're going to back calculate, prove it, okay, from basically right to left. And this is divided by the volume we put on the plate. And you may remember that this was 1 mil, okay, from each of them. So that was the volume you took from the dilution tube and you spread onto the agar plate to get those 100 colonies. Excellent. Do you guys remember what the 10-fold dilution factor was that produced that plate? It was the 10 to the 4th. So we're going to multiply that by 10 to the 4th. What we're saying here is we diluted it by 1 over 10 to the 4th. We made it a lot less concentrated. But we want a higher number. We need that original number, that big concentration number. So we have to multiply back our colony forming units per mil of that particular dilution tube times the number of dilution tubes we did. But not times the number, times the factors of 10 that we did. And in this case, that was 4, 10 to the 4th. So does this give us the concentration in the tube, which we're pretending we don't know, but we do know is 1 times 10 to the 6th CFU per gram or per mil? So let's do this. 100 CFU divided by 1 mil times 10 to the 4th is, I'm going to put 100 over here, okay? And I'm going to keep that orange. And then I'm going to add 4 zeros because 10 to the 4th is 4 powers of 10, which is actually 10,000. So we're going to multiply 100 by 10,000. And you can think about multiplying 100 times 10 to the 4th or 100 times 10,000 and putting that into your calculator. That's completely fine with me. Or you can remember the first video and add 4 zeros onto 100. Multiplying by 4 powers of 10 is the same as saying let's add 4 zeros to the end of whatever was in our parentheses. In this case, it's 100. So 100 with 4 zeros afterwards is 100 plus 4 zeros, right? You see that there? Good. And if we put that in scientific notation, that is indeed 1 times 10 to the 6th CFU per mil equals 1 times 10 to the 6th CFU per gram. So we can actually go backwards and prove it to ourselves. Do you see how we go from left to right? And we can predict which plate would have 100 colonies there between 30 and 300 when we knew the concentration to start. And then we could pretend we didn't know it at all and calculate backwards to prove it to ourselves using the concentration here formula finder. Outstanding job. Very, very good. Lastly, one more little thing. Now that we've gone through the example where we knew the original sample concentration and therefore the concentration in each of our 10-fold dilutions, We therefore knew the countable colony range of 30 to 300 was going to be 100, and we knew which plate that was going to be on. And we used that data at the end to calculate backwards and prove that the original sample had the concentration we thought. I want to show you one little variation. There are a lot of variations, but we're just going to do one. And so our little friendly yellow pipette tip is back to give you yet another pro tip. Sometimes smaller volumes are used for spreading than 1 ml. And so this is a dilution of a dilution. I hope your mind is intact. It hasn't exploded when I said a dilution of a dilution. And if it has, clean up the mess, pause the video, come back. I'm going to teach you what this means. We can put the brain back together, okay? Ready? Here we go. A dilution of a dilution. We are using, in this case, a smaller volume to spread onto the agar plates than what we did before. Okay? So, we already know our concentration in this first tenfold dilution is 1 times 10 to the 5th CFU per gram, or 1 times 10 to the 5th CFU per mil. But in this example, I'm creating a dilution of a dilution when I do the following thing. What if instead of spreading 1 mil, I spreaded 0.1 mils? And if you remember the first video where I asked you to convert milliliters to microliters a couple of times, you might recall that 0.1 mils is equal to 100 microliters, or moving the decimal place to the right 3 times, because there's 1,000 3 powers of 10 times the number of microliters per 1 milliliter. But in this case, that's not really what I'm wanting you to kind of pull out of here. 0.1 mils, or 100 microliters, is actually a tenth than dilution. It's a tenfold dilution from the 10 mils at the concentration of 1 times 10 to the 5th CFU per gram or per mil that I have in the tube. Prove that to yourself by doing the math. So take 1 times 10 to the 5th CFU per gram and make it 1 times 10 to the 5th CFU per mil. Multiply that by 0.1 mils, the volume you took out. Mils will cancel, but instead of having 1 times 10 to the 5th CFU, because 1 times 10 to the 5th multiplied by 1 is 1 times 10 to the 5th, 1 times 10 to the 5th times 0.1 is a 10-fold reduction. It's a tenth less. 5 powers of 10 becomes 4 powers of 10. You have 1 times 10 to the 4th CFU, not 1 times 10 to the 5th. See how the volume that I spread on the plate made those numbers change? Good, because you're going to do variations of this both in the online worksheet and in the in-class exercise. Let's carry this forward. The dilution series, that's not changing at all. It is still a 10-fold dilution series. We're still taking 1 mil and moving it from tube to tube that had 9 mils of solvent and et cetera, et cetera. So 1 times 10 to the 4th CFU per mil here, or per gram, is the concentration in the second tube. And again, we're taking 0.1 mils from it, which is a tenth. You're seeing it come through a little bit. Good, good, good. Okay. And therefore, 1 times 10 to the 3rd, that's still tenfold less than the plate to the left. You see how the tubes and the plates from the first to the second are still 10 times apart, right? The dilution series and the plates are still 10 times apart. But what I'm saying to you is, because you took a fraction of the volume you did on the last example, you went from 1 mil and spreading to 0.1 mils and spreading, you took a tenth less, then you end up with a tenth less colony forming units. That's going to be important here in just a minute as we finish this out. Okay, so if we keep going, 1 times 10 to the 3rd CFU per gram per mil, spread at 0.1 mils, you're going to get 1 times 10 to the second CFU. That right there, that's the colony forming units in the countable range. That's 100. And you might be saying to yourself, aha, I now kind of see where you're going with this a little bit maybe, because that is not the same dilution tube that gave me 100 colonies when I was spreading 1 mil from each of these things. Do you see how changing the volume you take from a dilution changes which plate gets to 100 colonies in the 30 to 300 range? Because there it is right here. But in the last example, we had to go one more tube past this because we were spreading more from each of the dilutions. We were taking more colonies forming units. We were taking more cells. Let's do that. Let's show you, okay? This is still the same concentration as it's been in our dilution series, but we're only taking 0.1 mils, and so that ends up with 10 colonies. That was the last number of colony forming units we had in our last example, where we were spreading 1 mils. But in this case, if we're doing 100 microliters from each tube, this ends up being 1 colony. So our countable range from left to right is slightly different. In fact, it's different by powers of 10. Okay? So this is the 10 to the third tube producing 100 CFU. And you might be wondering, if all that's right, if all that's true, then can we back calculate still to get 1 times 10 to the 6th CFU per gram? That shouldn't change, right? We have the same starting concentration. Can we prove it? Can we use our formula for determining concentration and make sure that this is okay, even if we just changed one thing, which was how much volume we were spreading onto the plate? I assure you this is the answer to this is yes, and I want to show it to you. Okay, so over here on the left, with everything here, right on the picture, let's do our math. Let's put the formula together. You may remember that in the bracket, we want the colony forming units in the countable range divided by the volume we spreaded onto the plate. So let's put that together. The colony forming units are still 100. That's the number between 30 to 300. But in this case, here's the two changes. Okay, the first comes in the denominator. We didn't spread 1 mils, we spread 0.1 mils. The second change was, outside this bracket, we have to multiply by the dilution factor. In this case, it's 10 to the third. It was the 10 to the third dilution, the 1,000th tube of where we started, that produced the 100 colonies. In the last example, from the same starting concentration, the same tenfold dilution series, we were spreading 1 mils, and so in the denominator we had 1 mil. And because we were spreading more and got more numbers on the plates, we had to go 1 power of 10 further. And so the dilution factor we multiplied by was 10 to the 4th. Here it's 10 to the 3rd. Okay? 100 CFU divided by 0.1. Well, that's the same as multiplying by 10, so you get 1,000. If you take 3 powers of 10 and add 3 zeros, well, you get 1 times 10 to the 6th CFU per mil, aka 1 times 10 to the 6th CFU per gram. You end up with the same exact number, and we can rest at night with a good night's sleep, because even though we changed the volume we spreaded to a smaller one, because maybe that's the only tools we had in the lab, we could still use our rules of how this works to get to the same exact number. And so we can put a bow on this or a ring on it if you like it. You can put a ring on it if you like it. You should. And we could head over to the online worksheet, do some math for ourselves, and we will reinforce this in class. Thank you again for being here, and great job.